Hello everyone. Today let us study about wilt disease in banana, which is caused by Fusarium. This is also called as Panama disease. The pathogen which causes this Panama disease is Fusarium oxysporum, subspecies Fusarium cubans. This disease was first recorded in Australia in 1879, Hawaii in 1904. In India, it was first observed in 1911. This disease is also found to be prevalent in Central, South America, Africa, Sri Lanka, Burma, Thailand, Indonesia, Fiji, Philippines, etc. Now, let us study about the symptom of the Panama wilt. As we all know, Fusarium is one of the pathogen which cause this disease, and it is one of the facultative parasite, and also it is one of the soil fungi. It's one of the facultative parasite which can be found even as a saprophyte and also being found as a parasite and causing disease. This Fusarium, if it is found in the soil, Fusarium oxysporum, if it is found in the soil during favorable condition, the propagules, they start germinating. The propagules can be either a microconidia, macroconidia, or a chlamydospore, or the hyphae or the mycelium, if it is found in the soil, may also cause the infection. Since this is one of the soil-borne diseases, usually the infection happens to the root system, and also this Fusarium causes systemic infection. That is, the infection can be found, that is, this fungi can get parasitized in the entire part of the plant. If you take any part of the plant, the infected plant, and you could just take a section, you can observe it under the microscope, you can find this pathogen in the host tissue, and this particular pathogen is found to be intercellular in between the in between the cells, host cells you can find. And these propagules which are present in the soil during favorable condition, they start germinating. They initially produces germ tube. These germ tube has the ability to infect the root system either by direct penetration or through wounds. If any wounds have been observed in the root system or in the suckers, the infection can happen. Once they enter into the root system, they enter into the stem. Here, in case of banana, the stem is a modified sucker. It's a modified sucker, as we all know. The stem is modified sucker where it can infect even these suckers. The target of this pathogen is the xylem. Xylem, you know, it's one of a water conducting tissue where they infect the xylem, where the infected xylem will be brick red or brown in color. However, the symptom on the plant can be observed only when the plant is around 5 months old. At that particular point of time, we could see the physical symptom, wherein which initially we can see the yellowing or the chlorosis of the older leaves. That is, the foliage or the leaves become yellowish because of the loss of chlorophyll pigment. That particular process is also called as chlorosis. And at the same time, these older leaves, this yellow streaks or chlorosis can spread into the entire foliage. The entire older leaves, they just start wilting, and later they just dry, as you can see in these images A and C. They dry and they just droop down. droop down and form a skirt like on the pseudo stem. As we could see in this image C, we can see the drooping of the older leaves which has already dried and are also showing some of the leaves which are showing the chlorosis. They just drooped on and form the skirt like structure over the pseudo stem. What are these pseudo stem? These pseudo stems are nothing but the leaf sheath, as you could see in the image just below the C. You can see the transverse section of the pseudostem. These pseudostems are nothing but the leaf sheath. The stem is actually the sucker which is found inside the soil. So this kind of symptom can be observed initially. Later, even the younger leaves also shows chlorosis. Finally, they wilt and dry. And if such infected plant, if you take a transverse section of the pseudostem and if you observe, you could see the entire water conducting tissue which is becoming brownish in color because of the decomposition of the xylem. As we all know, the Fusarium, once it infect the plant, their target, as I was mentioning before, is the xylem. They enter into the xylem tissue, that is, especially into the xylem vessels. Xylem vessels are the one which are mainly involved in conduction of water. They enter into the xylem vessels, they grow and develop inside that particular vessel and form a cottony mass, so that the blockage of the xylem vessel happen, and at the same, because the blockage of the xylem vessel, water conduction become difficult, so that the water is not supplied to the upper part of the plants. And at the same time, this particular fusarium has an ability of producing some of the enzymes like pectin methyl esterase, polygalactoronase, cellulase. These are the enzymes which are mainly involved in the decomposition of the cell wall. So they are mainly involved in the decomposition of the cell wall, so that the xylem which is present, xylem or the other tissues which are present, they just get decomposed, so that the xylem vessel collapse. That is how you could see the symptom, that is the decomposition or the rotting of the vascular bundles happen. Or at the same time, this fusarium has an ability of producing even certain toxins. Because of the production of toxin and also because of the blockage of the xylem, the water conduction become difficult, so that the transpiration is affected, because of which the closure of the stomata happen and all the other physiological activities of the host is being affected, which leads to the chlorosis of those leaves. That is how you could see the chlorosis followed by wilting and drying. And at the same time, you could see the longitudinal splitting. You could see the image towards the right hand side of this particular slide. You could see the longitudinal splitting of the older pseudo stem. So this is one of the characteristic feature of this Panama wilt. And another important symptom is the infection which you could find in the sucker or the rhizome, where the entire rhizome or the sucker gets infected. They just gets rotten, and you could see the odor of that particular infected sucker will be fishy odor can be observed with that of infected suckers. And at the same time, the fruits which has been produced out of such plants may not show much of the symptom. However, the quality of the fruit is not so great when compared to that of a healthy one. That point we have to note down in Panama wilt. There are some more organism which causes wilt diseases in banana. So for example, there are certain bacterial wilts also, which is caused by a bacteria called as Pseudomonas. Students, please make a note, Pseudomonas solanacearum, which causes wilt disease, that is called as Moko disease, M-O-K-O. Moko disease in case of banana is caused by Pseudomonas solanacearum, where the disease symptom is same as that of Panama wilt, where in case of mucormycosis, the fruits are also affected. There is one more disease which the disease symptom is like that of the Panama wilt, where the fruits are affected. If you take a section of the fruit and observe, they show some reddish discoloration. That disease is called as Shigatoka disease. Sigatoka. Shigatoka disease. Shigatoka disease, which is caused by a pathogen called as Mycosparilla musicola. Mycosparilla musicola is the pathogen which causes a disease called as Shigatoka disease. This disease was first observed in Java in 1902, and this disease is also found to be a serious threat even in India, which we can observe in Assam, Tamil Nadu, Karnataka, Andhra Pradesh, etc. So once again I will repeat, there are three important diseases with respect to wilt which we can come across in banana. One is Panama wilt, which is being caused by Fusarium oxysporum, whereas bacterial wilt called as moco disease caused by Pseudomonas solanacidum, and disease caused by Mycosparilla musicola, it's called as Shigatoka disease. Panama wilt, moco disease, and Shigatoka disease. These are the three important wilt diseases which we can observe, and this are very important for your competitive examination also. Once again, I will repeat the symptom. Initially, the symptom can be observed only when the plant, that is the visible symptom I'm talking about, can be observed only when the plant attains 5 months, where we could see the chlorosis or the yellowing of the older leaves, followed by wilting, drying, drooping over the pseudostem. And we could see the longitudinal splitting of the leaf sheath or the pseudostem. Older leaf sheath can be observed, as you can see in the images towards the right. And if you take a transverse section of the pseudostem and if you observe, you could see the decay of the leaf sheath also, and the rhizome or suckers are also being found to be decayed, and also you could see the odor is fishy. These are some of the important symptom of this disease. One more thing, the fruits are not affected, however the quality is not so great as with respect to that of the healthy ones, that you had to remember. Coming to the disease management of Panama wilt or Panama disease in case of banana, always prevention is better than cure. That is the reason use of disease resistant varieties such as Oven, Mongil, Rajabale, Vamanakili, Cavendish banana. Cavendish banana is one of the first disease resistant banana which was produced all over the world. Use of proper sanitary measures, that is, removal of all the infected part of the plant or the entire plant along with the sucker has to be removed, and it has to be burnt into ashes. And also keeping the plantation clean. Use of healthy planting stock. Care to be taken during cultivation, avoiding root injury and nematode control. Soil solarization, steaming also gives better results. Use of disease-free suckers for planting. Always see to that before planting, the suckers which we have obtained for the process of plantation has to be disease free. Avoiding contaminated soil, the soil, that is the land or the field which we are using for the process of cultivation should be devoid of any such pathogen. Crop rotation with paddy, application of lime 1 to 2 kg per pit to the infected pit after chopping of the plant's part. That is, after the infected plant has been removed from that field, even in that soil, the propagule may persist. That is the reason we can apply 1 to 2 kgs of lime so that we can control this pathogen. Dipping of sucker in carbendazim, 1.1% solution before planting. Before planting the sucker, better to treat the sucker with carbendazim so that the chances of infection can be avoided. Neem cake, as we know, neem is also one of a very good biopesticide along with Trichoderma viridae should be applied in planting pit to get the better results, and also soil drench with 0.2% carbendazim or rhizome or the sucker injection with 0.2% of carbendazim also gives a very good results. These are some of the method to control this Panama wilt.